It's network thermodynamic analysis of protein aggregation. And the presenter is Ashwin Vaidya, if I pronounced the name correctly.
You're muted.
Yeah, can you hear me now?
Yes.
Yes, we can.
And you can see, I think I've started sharing. You can see it.
Yes, go ahead. I will give you a sign, 2 minutes.
Sounds good.
Go ahead.
Some sound in the background. Okay, so thanks for organizing this fantastic event. It's a beautiful conference. I really enjoyed hearing all the talks. So I want to talk a little about some more recent work that I've been doing in the area of network thermodynamics. Okay. And its applications to protein aggregations. So before I start talking about that, I mean, I constantly changed my talk based on what I was hearing, and I wish I had submitted a different abstract because I could have told you a little more about some of the results that I've had in the area of maximum entropy production. I've been working in this area for the last 10 years or so. I'm a fluid dynamicist primarily, and I've been looking at pattern formation in fluid solid systems. And of course, we all know about the successes of convection in this area. That's an example that's often cited. But it appears, based on experiments and theory and some numerics that we've done, there are a whole bunch of other examples in this area, such as the case of the orientation of a symmetric body in a fluid. It's a question of pattern selection, or the shape of a deformable body, right? What final terminal shape does it take up? All of these questions, and also if you have multi-particle system that's flowing in a channel, there's a theory called effect called the Sigler-Silverberg effect, which tells you that the particles will aggregate at a particular location in a channel. And so all of these effects can be studied computationally and numerically and so on, and experiments have been done. But it appears, based on our computations, that the maximum entropy production principle selects these states correctly, right? So it identifies these from an infinite possibility of other configurations. So a previous work has been in the area of mechanical systems. The objective of this talk is slightly different. I wanted to see also, one objective was, can these kind of optimality arguments work in complex biochemical systems? And so I tried this approach on a different project that I was working on to see if, on Alzheimer's disease. So we all know about Alzheimer's disease. It's a neurodegenerative disease that progressively causes memory loss and thinking skills and language skills and so on. Now, biophysically, this is characterized by sort of aggregation of certain proteins. So there's a self-assembly process that's happening in the brain. So the actual biophysics of the process is such that you have these amyloid beta proteins that start aggregating, and the aggregation process is a classical sigmoidal, has a sigmoidal profile. So you have these monomeric molecules that keep aggregating and get larger and larger. They turn into dimers and trimers. Eventually they get to a nucleus. And post-nucleation, there is a rapid increase in the rate of aggregation until they eventually reach some sort of a stable fibrous structure. This single pathway we'll refer to as an on pathway. This is what is well known. But in realistic terms, this is, you don't just have a single pathway because of environmental factors. Reactions are highly stochastic. And so proteins are pushed into alternative pathways. There's multiple pathways in which aggregation happens. Particularly important to us is what we've been looking at, and this is my colleagues, biophysicists who perform experiments on this, and myself, a theorist, been looking at how the interaction between these amyloid beta proteins and lipids happen. So this interaction causes new sort of complexes, A beta lipid complexes to form that pushes the aggregation mechanics outside of the standard on pathway to some alternative pathway. And we're going to refer to this as off pathway. Now we make a simple schematic. So if I want to do any of this analytically, theoretically, I have to simplify the system. It's far too complicated. So I'm going to take a reduced order system, and we will look at two pathways. The one on the left that you see is what I'll call the on pathway. The one on the right is an off pathway, and they're mediated by the lipid concentration that is referred to as L over here. So A1 would be a monomer, AN over here would be a nucleus, and AM would be some large fibril or some large oligomer. And there are two bridges here, right? So the bridges will determine how the system is shifting from one state to another. And we only have two bridges because we did not put a bridge at the end because it's kind of known that post-nucleation oligomers are very stable, so they don't break up or transform that easily. This can be converted into a dimensionless system of differential equations. So it's a highly complex, even in a reduced order system like this, which has 6 species, you get a highly complex nonlinear coupled differential equation. This has all been non-dimensionalized. And you see these parameters alphas. These alphas, there are 12 of these constants. These are the reaction rate constants. 10 of these we determine from experiments, from fitting experiments. And the two which are colored, alpha 3 and alpha 4, these are what we call the forward bridge parameters. So these are essentially the measure the lipid concentration in the system. And they will tell us tell us about how the system switches from one pathway to another. Okay, so we take a game theoretic approach to this problem. So we're playing a game between the on and the off pathway. And I'll show you some computations. And we have to assume a few things. We take our bridge values to range between, these parameters to range between 0 and 2. Everything is non-dimensionalized, so it's, the ratios are chosen in accordance to that. The default initial condition is chosen so that the initial concentration of the monomer is set to 1 and everything else is 0. But we also consider other cases, which we call seeding cases, where in the initial state, you can have multiple other species, multiple concentrations of other species present. Right? And seeding also is a very interesting thing. Experimentally, seeding is used as a way to control the mechanism. Right? So for a given initial condition, what we want to do is determine which of these so I'll think of B1 and B1 prime, Bn and Bn prime, Bm and Bm prime as being like species in the on or the off pathway. And we want to determine the conditions under which these like species win. So when does B1 win? When does B1 prime win? And so on. And so this results in the creation of, or identification rather, of several network topologies in the system. For this particular simplistic system, there are 8 different topologies. Right? And network pathways, let me call them. And these are the dominant pathways. So the one that you're seeing on the top left, so this would be the 1, A1, or B1, Bn, the nucleus, and the Bm. So this is the first one on the left, which I'm calling gamma 1, is telling me, we have different ways of labeling it. This is also called the on, meaning at every stage, the on pathway species wins. Or the second one will have FOO, which means the off pathway wins, and then the on pathway wins, and then the off pathway wins, the on pathway wins, and so on. So there are 4 on top that you see that all end up in the on pathway terminal state, right? And this is what we're calling the safe state. It was initially thought that forming fibrillar plaques or finding them was indicative of some sort of toxicity in the system. But now it's realized that it's the smaller oligomers, right? These are called insoluble oligomers in the brains that sort of result in the neurodegeneration. So it's really the off pathway which is not safe. At the bottom, and the top ones are colored green, light blue, yellow, right? Light blue, dark blue, and yellow. The bottom ones are reds, lavenders, and black. those always end up in the off pathway state towards the end. No matter how they start and no matter what it is in the middle, they always end up in the off pathway towards the end. And these are what we'll call pathogenic states. Right? So there are 8 possible network pathways over here. Dominant pathways. This is not saying this is the only reaction that's happening. They could all be simultaneously happening, but we have 8 possible species over here, 8 pathways over here. Now what we do is we solve the differential equation, the system of differential equations. We look at the equilibrium states of these under various conditions, meaning under various values of alpha 3, alpha 4. So you have a phase diagram over here, which tells you under various lipid concentration, which sort of topology seems to appear or dominate in the system. Right? So you'll find that when alpha 3 is low, alpha 4 is low. The picture on the left shows you that the green state, which is the on, that pathway will dominate the system. It could mean that the others exist, but that this is just much more powerful, much stronger. Right? And as alpha 3 increases, this goes from the color changes from green to the light blue, and the light blue would be the off, on. So it's shifting as alpha 3 changes, you're going from this particular state to this particular state. And the picture on the right simply calculates the percentage, the fraction of the various pathways, right, under these conditions. And so this picture was shown just for the standard initial condition where you only start the reaction system with a certain concentration of monomers and let the system evolve. But now if you start to see the system, you also find some interesting phase diagrams here. And in particular, so look at the one on the left, right? The top 2, in fact. This is if I seed this with an on-pathway nucleus, then what I find is the red and the lavender ones are the pathogenic states, and those are let's call them dangerous states. We don't want them. And it seems like if you seed the system, if you change the initial conditions, so you have monomers as well as some of the nuclei, then you sort of get rid of the so-called dangerous states. And the same with BM. And this is experimentally well known that seeding can help, and the right kind of seeding can actually serve as an attractor for the system. Right? On the other hand, seeding with off-pathway species increases the likelihood of the red and the lavender regions, meaning the off-pathway reactions to dominate. Okay. And then this is, for sake of time, I'll just skip this. So now the main question that I want to ask is, well, thermodynamic arguments have helped us select the right kind of configurations out of, in complex systems before, in dissipative mechanical systems before, but can similar principles be applied to understand the configuration over here? I'm thinking of these 8 different states as 8 possible configurations of the system. Can a maximum entropy production principle work here? I'll tell you a little bit about that later, but what we started with is trying to look at the Gibbs free energy of the system. It's a standard procedure in chemical reactions to analyze the delta G's in the system. And delta G is related to the chemical affinities of forces. And so we calculated the delta G's for each of these 8 pathways. Now, each of these 8 pathways is composed of subpaths. These networks that I showed you, the 8 networks, it's not a single reaction. It's a collection, it's a union of 4, 5, 6 different reactions. So we compute the net force in each of these pathways. And then we look for I'll show you some of these profiles in a second. The negative, if the Gibbs energy is negative, it tells us that the reaction is spontaneous and stable. If it's positive, then it's not, right? So this is what we're looking for. So what we do is we take one of these phase diagrams and we take different cuts along these phase diagrams. So take this cut over here, alpha 3 equals 0. So, and see, okay. So this is telling us, that if I go along this cut, alpha 3 equals 0.
5 more minutes.
Okay.
5 minutes.
And so if I take this and I vary my delta G calculation along this path here, then the only reactions where delta G is negative, right, happens to be the green and the lavender, which is identical, which is almost exactly, we've left out the yellow for some complicated reason, which I don't have time to go into. They also do show up. But you see that delta G sort of identifies, not all reactions are happening here, right? Only two of these reactions are happening, right? And these are the stable ones. And they seem to agree with what we see in our phase diagram. Now, if I change this to alpha 3 equals 2, so I'm taking a cut of this phase diagram along this direction, then I see lavender and red to be the only two negative delta G reactions pathways that have delta G negative, right? Which means these two are spontaneous and stable, the others are not, right? So in a manner of speaking, delta G, not a manner of speaking, it actually is helping us identify stable reactions. Not every steady state is realizable, right? Not every steady state is physically meaningful. So without getting too much into this, I'll simply say, so we do this for different cuts just to make sure that it is telling us the right thing. And absolutely, so no matter which cut of this plane we take, we find delta G to be telling us exactly when, close enough to be telling us which reactions are stable, which are not. Okay. We also did the same thing for a few other cases because we wanted to make sure that for any phase diagram, no matter what the initial conditions are, what sort of seeding conditions we have, that delta G just does work. There's no way to prove this in general as a theorem. So all in all, the significance of the thermodynamics here, it is quite significant. It's very important. Like I said before, the kinetic equations will help us determine the mechanics of self-assembly and self-organization. But they don't tell us about which ones are physically possible, like which are realizable, which are feasible. Minimum delta G over here seems to give us more information. So minimum of the Gibbs free energy. And I'm not sure if there's I've heard of different kinds of free energy principles, and I'm not sure if these are exactly the same thing, if they're always measuring the Gibbs free energy or other kinds of free energies. Entropy production, we've also tried to calculate the entropy production for these different pathways, but we haven't had success here. And my feeling is that some of the only sort of negative papers that I've found that reject entropy production seem to come from chemistry. I think it's a little more complex in chemical reactions to prove this point, and I have a feeling that this could be because you're not looking at a distributive system, and the model only shows you the chemical species here. It doesn't show you the environment, so that has to be modeled as well. So there is something going on. You have to look at the whole system, and we're not looking at the entire system. Another interesting aspect of of this particular calculation is that we find certain what we call surrogate states. So you can see that some of these delta G profiles go hand in hand. The green and the lavender will always go together. Right? So we're trying to build a slightly more interesting theory, a category theory for that sort of tells us a little more about the true complexity of the system. The system, even though I've identified 8 of these states, it's a little more complex than that. So final thoughts, I find a lot of connections between some of this, the discussions we've been having over the last day and today to the work of Dilip Kandipudi and his group at UConn, his collaborators at UConn, and the work of Adrian Bejan and constructal theory. In fact, he used this argument, constructal argument, to show sort of orientation of bodies in one of his earlier works. And it's very interesting. I hope to have more conversations about many of these things. So, I'll stop over here and just want to acknowledge my co-authors, my student, Joseph Pateris, who just graduated. This work is part of his thesis. And this work was supported by NSF. So, thank you very much. I don't hear you, I'm sorry.
Seamus, you're still muted.
I was trying not to create noise, and I created no information whatsoever. I was saying that we can save the questions after the end of the next talk.